obviously, different technologies are emerging. No, there's no clear winners yet. Looking longer term, what do you think would make or break a company in this field? What will be the key component?
I still think the breakthrough going to be coming from different technology platforms, not only one, but some of those may be a little bit forefront than others. I think nanoparticle clearly has better chance to reach to this goal. GERD and some liposome-based also have great potential, especially the liposome-based delivery system already attract a lot of attention due to the long time study background on gene therapy and other traditional drug delivery. So liposome is forefront currently. However, liposome also face some scale-up issues and other issues, especially related to the potential toxicity, because most of the liposome we're using is a cationic liposome, it's positive charged. It may have certain inherent issues we need to overcome. On the other hand, nanoparticles use either liposome-based or polymer-based or metal-based, give you a lot of diversities. You can manipulate easily, and we know it's a relatively smaller size. You can also control the charge, so give you a quite a broad spectrum for the flexibility you can work with. We're currently working with 7 different types of nanoparticles, and we believe at least a couple of them already ready to be in the clinical testing. So we are currently moving our program into the late stage preclinical development. Hopefully next year we can move into clinic testing.
Okay, now assuming we solve the delivery problem through nanoparticle or whatever other methods, is there an inherent danger or greater risk now that you're getting into the cells for medicine, for drug development? Are there off-target effects or higher toxicity, whatever?
Very good. The question. I think any drug may all have the concern about off-target effect. Not only SIR drugs. So we know other drugs has potential toxicities. We need to have more human testing really answer that question. We also know some basic material. What's their possible toxicity? What's their possible adverse effect, so we can well plan our studies to address those issues before we really move into human testing. For example, RNAi, if RNAi potentially can integrate into the human genome, although we don't want them really go to the nucleus, but it potentially may happen. And also the off-target effect may cause some, we call it exaggerated pharmacology. That means unwanted efficacy. It's too much drug activity, so on. So that's all has to be addressed, both based on our knowledge on those biology understanding, and also we need to test in human trial.
Okay, so Sirnaomics is currently, you said it's preclinical, but hopefully moving on to clinical next year.
Yes.
What are the data look like so far that you have, and what are the different targets?
Okay, we are taking quite a unique strategy. We are using multi-targeted siRNA cocktail to treat those diseases. We believe due to the homogeneity of siRNA, using a multi-targeted cocktail going to be more effective. That's the advantage other drug modality like a monoclonal antibody or small molecules may not have. So we should take advantage of this homogeneity of siRNA in terms of chemistry. At the same time, we also understand the delivery issue is a long time issue. Even for small molecule drug, you still have delivery issue not overcome. So we understand the difficulty of certain delivery issues. So we go after a relatively low-hanging fruit approach.
Such as
wound healing program. There's less hurdles to address for delivery issue because it can be easily delivered into the wounds. It's already opened, and it's has tremendous unmet needs, especially for we call it scarless wound healing. So I think for ladies like you may be really interested in those kind of topics. And another one is for influenza treatment. We know H5N1, H1N1, it's become more threatening to our world, to almost the entire humankind. So if these two get together, mutated, and acquire their harmful features, going to be really bad for us. So we try to develop novel therapeutics to address those issues. Use multi-targeted siRNA, target the conservative region of those viruses. We hope we can develop drug against any possible resistance due to the resorption, mutation, and other typical influenza viral mixtures.
Oh, so how does that work? You these multi-targets, you apply for approval at the same time, simultaneous? How does?
Okay, I know where you are coming to. It is a question whether FDA will finally pass this kind of strategy. We know FDA is very keen on multi-targeted approach for oncology, for antiviral. So this is nothing new for them. They really encourage us to do that. We have interaction with FDA. I believe for antiviral and anti-cancer application, we will have very easy way to go through it. For wound healing, we still need to communicate with them, see what's really going to happen. But I'm quite optimistic about all those issues. But as any scientific endeavor, you need to work hard on the real science. Same time, when you make a drug, you have to go through the regulatory bodies to make sure your thing is safe.
Sure. So we've had this conference yesterday, and it's in its second day today. Have you heard any interesting progress among colleagues that you think it's worth noting in the field?
Actually, yes, there are several things quite exciting. Although some of those we already learned from latest news, I'm pretty much on pulse of what's going on in the field. We know GERD works very well, just published a couple of months ago in Nature, used this novel glycol-based system for oral delivery. That's really amazing if we can do that. So there are tremendous applications. Although right now still in its infancy, so we want to see what's going to happen, especially go to human testing. Another interesting part is how to take advantage of mRNA as either therapeutic agent or as a target of siRNA therapeutics, how to avoid the potential off-target effect, or even take off-target effect as a benefit for your therapy. So that's the discussion, also very interesting. We had a roundtable discussion yesterday. We try to address the issues along different lines for delivery. I think scientists, at least in the room, have full understanding what we need to do, what we already know, especially for the preclinical stage. So right now, we all try to learn more about if we move into clinic in human, what's going to happen.
Are people or companies collaborating, or is it just more kind of a learning best practice kind of thing? Is it more informal, or are there formal collaborating efforts?
There are a lot of collaboration ongoing, and there are several CEOs in this conference. Actually, we may have some deals signed up still in private, but this is another good forum to allow colleagues exchange ideas and allow companies for potential partnerships.
Right.